there are a lot of bottlenecks in the early bioprocess development in the upstream processing. There is where you do all your strain development, trying to find the best media, setting up those initial culture conditions to get the best growth and productivity. And very often see things get stuck in this early upstream phases. And if you're not doing enough comprehensive screening and optimization right after the beginning, like with your strain selection or media development, you're often setting yourself up for suboptimal titers for product quality. And those issues become way harder and more expensive to fix later on when you are further downstream or trying to scale up, for example.
Welcome to the Smart Biotech Scientist. Are you struggling to choose the right screening approach for your early stage bioprocess development? Wondering if you're creating bottlenecks that will haunt you downstream? Today, we're tackling the critical decisions that separate efficient process development from costly delays. I'm joined by Sebastian Blum, who is a microbiologist with over 2 decades in life sciences and market development manager at Beckman Coulter. We're diving into high-throughput screening strategies that actually work. And thank you to Beckman Coulter for sponsoring today's episode. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Sebastian. It's good to have you on today.
Hey, David. Fine. Now happy to hear. Thanks.
Sebastian, share something that you believe about bioprocess development that most people disagree with.
Well, that's an interesting one. So I think maybe the James Bond question, or at least I call it the James Bond question, which often arises in the community. Is it stirred or is it shaken? And in my opinion, in the early screening phase in R&D of microbial cultivations, it's not about how the oxygen is introduced, but how much is introduced. So what is the KLA and what is the oxygen transfer rate? So in my view, for this question, at that stage, it's irrelevant whether it's stirred or shaken.
You have quite a long career already in bioprocess development. Take us into your story in the very early days. What drew you into this field and what were some interesting steps along your career path?
Actually, I've been in the life science industry for over 25 years now. My background is in biology with a focus on microbiology. Back in, I think it was 2010, there was a small company in my network which really caught my eye, and they had invented a micro-fermentation system, and they were really focused on using it for bioprocess development across different markets. I was super hooked, so I started talking to potential customers, just asking what they thought about having a tool like that in their lab, and their feedback was quite positive, and that totally convinced me at that time and led me to join the M2P Labs team in 2011, which was then acquired by Beckman roughly 10 years later. It was, at that point, it wasn't an easy decision for me because I just had my first child, and honestly, I was looking for some stability at that time. But in the 14 years since I have been there, I've never regretted the choice a single day.
I'm curious, Sebastian, because you work with a lot of different companies. Please paint us a picture of how certain biotech companies approach process development differently.
Okay, that's maybe really interesting because when I look at biotech companies, they actually tackle process development quite differently and often depends on who they are. What do I mean with this? For instance, if you got your smaller startup or those academic spin-offs, they're usually all about speed and keeping costs down. So they might start with a simpler solution or lower throughput methods, things like shaker flask, for example, just to quickly prove their concept. And it's all about getting that initial data very fast. And then on the other hand of the spectrum, you have the big established pharmaceutical companies, and they tend to invest heavily in really robust, data-rich early phase development, for example. They are using advanced automation, high throughput systems like the BioLector right from the start. The main goal is normally to de-risk also the process as much as possible, ensure it's scalable afterwards, and build this super comprehensive understanding of all the critical process parameters. Why are they doing this? Because they have got the meat to meet the stringent regulation requirements down the line. And then, of course, you have the CDMOs, those contracts, development and manufacturing organizations. They really have to be super flexible, offering a whole spectrum of approaches simply because they are serving so many different and diverse clients, each with their own unique needs and at different development stages. One common thread that I've seen among all these successful companies, regardless of their size, it's the commitment to generate good data and doing it early and often. Hope that answers your question.
Just following up on your answer, because this is very insightful, what is the main difference between the small and the large companies? Is that mainly cost, or is it also expertise or the ultimate strategy? What is your observation?
No, I would say also costs very often, but sometimes you're underestimating the startups where strong investors are, for example, included, and they want to push the startups quickly to the market as possible. And then all of a sudden they have also quite a high budget. But yes, of course, startups normally have less budget and less resources compared to big pharmaceutical companies, for example.
Among the projects you are involved in, where are the most common bottlenecks or challenges?
I see that there are a lot of bottlenecks in the early bioprocess development, in the upstream processing. There is where you do all your strain development, trying to find the best media, setting up those initial culture conditions to get the best growth and productivity. And very often see things get stuck in this early upstream phases. And if you're not doing enough comprehensive screening and optimization right after the beginning, like with your strain selection or media development, you're often setting yourself up for suboptimal titers for product quality. And those issues become way harder and more expensive to fix later on when you are further downstream or trying to scale up, for example.
This is an excellent point, Smart Biotech Scientists. Please listen to this again. I'm going to repeat it. You need to focus on it early on, because if you make mistakes early on, these can become very expensive. So thank you very much for saying this, Sebastian, because this is also a message I try to repeat as much as I can, and very important advice. If you look now at early stage bioprocess development, what are the most common misconceptions you come across working with a diverse group of people?
I think most scientists are aware of the particular importance of the early screening phase. So I wouldn't call it a misconception. However, I more often see scientists screening in, for example, batch mode, even though it's already foreseeable that the final process will run in a fed batch mode, so including pH control. In my experiences, this can lead to an incorrect ranking, which then only becomes apparent during the scale-up process. This result can result in a significant loss of time, and as you essentially have to start all over again afterwards. I mean, this can be accounted for in project planning, but why not directly screen in the mode in which the final process will ultimately run afterwards?
Yes, this is important to know what will be the system your process will be running in. Or in other words, start with the end in mind. This is important, and then work everything backwards, right?
Yeah, exactly.
Now, early stage development can be quite overwhelming if you have never done it before, especially if you're launching your own startup. You have a lot of things to look after. And now, since there are so many different bioreactor systems, how should a founder go about, or even a more advanced biotech company, because you have very different systems. So perhaps let's start like this, Sebastian. Can you paint us a picture of the various small scale systems that are available, and perhaps just give us the 2-minute version of when should you work with one or the other system?
I would say when scientists are looking at bioreactor systems today, it's like they are choosing from a pretty clear spectrum. And I would say each option has its own trade-offs. You can start, for example, with the shake flask. They are super cheap, easy to set up, and good for just rough initial screening and checking viability, for example. But That's the con. The process control is terrible. pH, DO, temperature, they are all over the place. You get limited real-time data. Reproducibility isn't great, not to talk about scalability. Plus, if you have a lot of samples, it's really lab intensive. Not to mention the cleaning and potential cross-contamination. So there are, of course, fields where you can use them, but you need to really check carefully when to use them. Then, for example, if you look for benchtop bioreactors, 1 liter to 10 liter stirred tank bioreactors, for example, advantage are you get excellent process control. They are great for actual scalability studies, and they are much more representative of large-scale production compared to, for example, the shake flask. And you can get really rich kinetic data from them. But downside is also they are expensive per experiment and your throughput is normally low. You can only run a few in parallel, which takes a lot of lab space as well. And there's still a lot of manual work involved and they are just slower for extensive optimization. So these are, in my experiences, ideal process characterization, initial scale up once you have already identified your key parameters. And then finally, the high throughput platforms, the advantages, the pros is this is where you get massive parallelism. We're talking about dozens of experiments all in parallel, and you get advanced process control, even at the micro scale with real-time, non-invasive measurement for things like optical density, pH, dissolved oxygen, different fluorescence proteins, and they are reproducible and generate tons of rich data for DOE studies, for example, really speeding up early stage optimization. But also there we have disadvantages. The initial capital investment is higher normally, and you really need good experimental design and data analysis skills to get the most out of them. So these are best for, in my opinion, rapid strain screening, media optimization, and doing really comprehensive process development in those early and mid-development phases. The big takeaway is, I think, you've got to pick the system that truly aligns with your development stage, how much data resolution you need, your throughput requirements, and of course, your budget. And often companies will use a combination of these systems in a phased approach, for example.
Basically, we have 3 different main types of bioreactors, correct? We have the shake flasks or the spin tubes, which are very simple in handling, but you don't have a lot of control over. We have the bioreactors where obviously the throughput is not very high, but you have a lot of advantages because you can control these bioreactors. And then in the middle, we have all what we call these high throughput systems where you can do a lot of screening. And you can do, to a certain extent, also some process control.
Definitely. So the bioreactor really stands out quite a bit, both for how easy it is to use and the quality of data it gives you, especially compared to traditional methods and even some other automated systems out there. So usability-wise, it uses a micro-titer plate format, which is well known and easy for high throughput work. And setting up an experiment in the BioLector software is intuitive and allows the user to monitor in real time and visualizing all your data. Plus, it has this non-invasive optical measurement technology, meaning you don't need to have to pick probes into individual wells. That just makes handling so much simpler, reduces contamination risks, and also lets you run it continuously unattended. It's a big advantage. It cuts down on manual labor massively compared to shake flasks or even a lot of benchtop fermenter systems. Data quality, it gives you real-time online measurement for critical parameters like biomass, which is measured via scattered light, pH, dissolved oxygen, and fluorescences, for instance, for GFP expression. And it does this with time difference of just a few seconds. So unlike systems that just take occasionally offline samples, the BioLector captures all those dynamic changes during a much richer kinetic profile of how your microbes are behaving under different conditions.
How does the BioLector differ for, let's say, an ambr 15 or 96 depot plate? I imagine a lot of you listening, you have worked with either one of the systems. Can you just tell us what are the different volumes? How many conditions could you typically test? And what are some main differences or also some commonalities between the 3 systems?
The BioLector system is using working volumes from 800 microliters up to 2.4 milliliters in a 48 well format. The 48 wells are batch fermentation. If you want to go for fed batch fermentation with seeding, it's 32 experiments in one single well because we need 16 reservoirs in this plate for carbon source and for adjusting the pH. So compared to ambr systems, 250 milliliters, there is, of course, a difference. We are measuring the pH with so-called optodes, optical sensors, in each of the wells individually. By this, we also don't need to take any samples, for example, for biomass measurement, which we also try to avoid, of course, because the volume is not that high. And that can also be a disadvantage if, for example, the higher volume is needed. Then I would say ambr system with 250 milliliters offers much more possibilities than an 800 or 1,500 microliter reaction run. I would say some of our customers are using, for example, the BioLector also to reduce or de-risk their ambr 250 runs. So that means they're using both systems in combination just to get a better selected choice into the maybe more expensive ambr runs to de-risk their experiments.
Can you run perfusion at all in the BioLector? Or at least I'd say...
Sorry, no, that is not possible. We cannot run a perfusion in the BioLector.
Okay, understood. And what are the cell types that are best suited for the BioLector?
BioLector is developed for microbial applications. So everything that needs high oxygen, changes the pH quickly, that is made for BioLector. Definitely not use mammalian cells in the BioLector. We tried this in the past because we were curious, but the results were quite poor. So we said, okay, that's not our target market here. But the possible microorganisms that can be used reach from strictly anaerobic applications to phototrophic organisms, fungi or filamentous fungi, even bacteria, yeast, E. coli, Pichia, Saccharomyces, Bacillus, you name it. So it's very flexible in terms of microorganism cultivation.
Can you give us some specific projects or cell types you have worked on recently?
We are working with Pichia and the methanol induction, which is very common in the industry as well and gives a big advantage. But also lactobacillus, for example, in the food industry is often used with BioLectors and low pH sensors that we are offering because of the low pH, the lactobacillus where it is cultivated. So I would say these are some strong organisms that we are working together with customers at the moment.
I'm just curious because what I am hearing, Sebastian, is that the BioLector excels at screening experiments because you have 48 wells you can use for media screening, for different culture conditions, and now giving different approaches, especially modeling approaches and so on. I mean, there are smarter ways to define your experiments and there are not so smart ways to define your experiments. Let's say like that in a very simple way. So I'm just curious, how do you guide companies also to make the best use of the BioLector? Can you give us some examples? What companies typically need, or are they very experienced and they know exactly what they need to do?
Typically, when they got in touch with BioLector, we normally are doing also demonstrations of the system. And at that point of time, we understand what customers need and the customer understand what the system can offer to them. So that helps a lot in the process of seeing whether the system gives an advantage to the customers at all. But of course, there's an intensive training because not everyone is very well familiar with high-throughput screening systems or with the BioLector specifically. And also software is, of course, new. So there's normally a training of 2 days, 3 days of application specialists involved where we go through what customers want to see and also what kind of complexity there is. Our system allows, or the BioLector allows, a scripting that means you are able to do some more complex protocols. And these complex protocols done in Lua. There you need to have somebody on board is able to script in Lua. Of course, that is not always the case. And we have also developed a user interface that helps you doing that. But that's very often we face, especially in the industry, less in the academic areas. We are facing customers who are not able to do Lua scripting on their side. And therefore, Beckman is also offering this to be done by us. So we can get in touch with customers and talk about what they need to be able to program this Lua script, especially for them. I would say from the biological part, they are very experienced normally. But of course, there need to be training on how they could use their knowledge on the BioLector most effectively.
That's it for part 1. We've covered critical ground on process development strategies and early stage decision making. In part 2, we'll continue exploring screening technologies and practical advice for maximizing your data quality. If you're finding value in these insights, please leave us a review on Apple Podcasts or your favorite platform. It helps other scientists discover practical advice like this. Thank you so much for tuning in, and I'll see you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, Visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.